Hello, we just learned about carbohydrates, monosaccharides linking together to make disaccharides or polysaccharides. Now we're going to talk about amino acids, which are also another biochemical building block. These are what make up our proteins. So amino acids are generally very simple structure in that they all have a very common structure of a central carbon, which we call the alpha carbon. And then attached to that alpha carbon are different groups. We have a carboxyl group, carboxylic acid. We have an amino group, or an amine. Okay. We also have a hydrogen. And then our fourth group attached is an R group of some kind, so variable group. All right, but they all have this kind of structure. And on the right here is a little nod to L-glyceraldehyde. And you can see that these amino acids are a little bit similar, right? Like look at serine here, CH2OH, we have an H. The only difference between these two are we have a carboxylic acid instead of an aldehyde. And then we have an NH2 group instead of an OH group. So, but very similar. Now, it turns out that there are 20 amino acids that are very common. And so these are the ones that you would most likely memorize in biochemistry, which means you'd know the structures, you'd know their codes, their abbreviations. These are all right here on the next page. So I remember making flashcards in biochemistry class with the structure of each amino acid, and then you had to know the name was glycine. It had an R group of a hydrogen. It had a 3-letter symbol, GLY, and a 1-letter abbreviation, G, capital G. So all of these amino acids are ones that you would ultimately need to learn if you were taking biochemistry. So with those amino acids, there are 10 of the 20 that are called essential amino acids. And that means that we need to obtain them through our diet because our bodies can't make those particular ones. And so those are these listed here. So now, you don't need to memorize these amino acids. You'll be able to use your chart, but just for practice, just getting used to thinking about some of the names. I think you've heard of some of them as well. It was just Thanksgiving last week, and at least at the time of this filming, and a lot of people say there's a lot of tryptophan in turkeys that makes you sleepy. So here's tryptophan, and that's in the protein, which turkey is high protein food. So I've read articles where they say it's not just tryptophan, though, it's the serotonin or the that satiated feeling that you have from eating. But anyway, cyclic structure, we see that there are several with cyclic structures. Tyrosine has a ring, phenylalanine, tryptophan has 2 rings, proline has a ring as well, it's not aromatic. Histidine has a ring as well. Okay, you can see some of them are aromatic, some of them aren't. Aromatic side chain, basically all the ones I mentioned except proline, I think, are aromatic. We have side chain with a basic group. Notice that some of the amino acids have nonpolar side chains, such as alkyl chains. Some have polar side chains, and some have acidic and basic side chains. So here's 3 that have basic side chains: lysine, histidine, and arginine. All right, sulfur atom. There's 2 with sulfur. Methionine is one of them, and cysteine is the other one. And so those are good to know when you're doing research with proteins, or you're studying proteins a little bit more. You know how cysteine can be oxidized or reduced, and we're going to start talking about disulfide bridges and things like that. Acidic groups could be like carboxylic acid groups on the end here, glutamic or aspartic acid. And a proton that can hydrogen bond, there's several. Serine here, threonine, and tyrosine are all examples of that. Okay, but you also have the carboxylic acid ones. And any of these amines can also hydrogen bond. So one thing that's very important to know about proteins is how they fold, and that has to do with their function a lot of times. So their structure is very important to understand. Their structure is based on the charges. So if I have an amino acid, let's say that there's two regions where there's charge. So for example, there's the carboxy end, which could be neutral, or it could be in the acid form or its conjugate base form. And then I also have the amino group, which can be in its conjugate acid form or its base form right here. All right, so I have two different groups that are sensitive to pH. Now, each of these groups can be thought of as, you have your acidic site and you have a basic site. Sometimes the acidic site turns into a basic site, or a basic site turns into an acidic site. What's important is to recognize that there's an acidic and a basic form for each part of this amino acid, for each functional group. Now, back in the day, probably in Chem 163, you learned about the Henderson-Hasselbalch equation. So that goes something like this, if you recall. The pH of a solution is the pKa of the acid plus the log of the conjugate base concentration over the conjugate acid concentration. All right. Now, don't worry, we're not going to use this for calculations, but we are going to use it sort of in a qualitative way. And to do that, remember, there's a very significant thing that happens when these are equal right here. When these are equal, this equals 1, and the log of 1 is 0. And so pH and pKa equal each other when I have a 1-to-1 ratio of a conjugate acid with its conjugate base. Okay? So what that means is, let's say that I have a pKa of a carboxylic acid of an amino acid. So the pKa of this hydrogen here, let's say that it's generally about 2 to 3 pKa units. And so what does that mean if I have a pH of 7.4? Now what that means is, if I was at a pH of 2 to 3, so let's say if the pKa is 2 to 3 and my pH is 2 to 3, if my pH was 2 to 3, I'd have an equal chance of being deprotonated or protonated. It'd be 50-50, right? And that's because of this Henderson-Hasselbalch. So in other words, in a 50-50 mixture of conjugate acid and base, the pH is equal to the pKa. So if this is 1-to-1 at pH of 3, what happens when I suddenly increase the pH to 7.4? That means that I'm getting more basic in solution. So that means I'm not likely to have as many H's, and I'm more likely to have this deprotonated form. So right here, when the pH is greater than the pKa, the basic form predominates. And usually we want to see a difference of at least 2 units, meaning at pH 2 to 3, it's going to be about 50-50. At pH 3 to 4, it's going to be maybe 10 times more, because remember, this is a logarithmic log base 10 scale. When I get to be 2 pKa units different between the pH and the pKa by at least 2 units, that's 100 times difference. So that's going to give me a major form. So at a pH of 7.4, that's many times greater. That's like 10 to the 4 times 10 to the 5 times greater, right? So that's going to highly favor the basic form. So the carboxylic acid then is deprotonated when we're at a pH of 7.4. All right, now let's try the next one. pKa of an amino group is about 9 to 10. So for an amino group, right here, this is 9 to 10. So at a pH of 9 to 10, I'm 50% this way and 50% that way, okay? But if I'm at a pH that's lower, so 7.4, am I going to be more protonated or more deprotonated? So using the same kind of reasoning, if I'm 50-50 at pH of 9 to 10, but now I'm going more acidic, right? So more acidic than the pKa, then I'm going to favor the acidic form. And the acidic form is the protonated form right there. All right, so far it turns out that I'm going to prefer at a pH at 7.4, which is physiological pH or blood pH, we're going to favor the deprotonated form here and the protonated form here. So this is the form most amino acids will be in at a physiological pH. They'll have a carboxyl group with a negative charge on the O and a positively charged N. So this is a protonated amine here. Now, when you have different charges, when you have a molecule with opposite charges in that molecule, this is called a zwitterion. It's the zwitterion that is the prevalent form at physiological pH. Okay, most carboxylic acids have a pKa of 5. Can you think of a reason why amino acids have a pKa of 2 to 3? So for this problem, you might want to think about how stabilized this negative charge is. And the more induction I have, the more spread out that charge can be. So remember, an oxygen doesn't want to hog all the negative charge. It wants to spread it out. It can spread it out through resonance. It can also spread out through induction. So by having the nitrogen there, it helps to pull the charge and spread it out more, which delocalizes that charge. And that stabilizes it. So I'm going to say the amino group stabilizes the conjugate base by induction. It pulls the charge away, therefore spreading it out. All right, so for this one, basically just look up the amino acid alanine. So it has a pH, it's in pH of 10 solution. Here's alanine right here. Luckily, this does not have a basic or acidic group here, so it's just this group and that group, and they're in their zwitterionic form. So alanine, I'll draw that. Alanine is a carboxyl with an alpha carbon, which is a hydrogen and a nitrogen. And I'm not sure what these are yet. I see I don't have a physiological pH, so I have to consider that might be different. So if this is a pKa of 2 to 3, and I have a pH of 10, that's really basic, right? So I want the basic form of this. So I'm going to take my H away. I'm going to make the basic form. This guy has a pKa of 9 to 10, and I'm at 10. So that means I'm 50% protonated and 50% deprotonated. So in that case, you can just decide, or you can write both if you want. So that's the idea is, what does the structure look like? Is it charged? Is it neutral? What parts are charged or neutral, depending on the pKa of each group and these pHs. Now notice some of these side chains have pKa units themselves. So tyrosine, let's look at that real quick. Tyrosine has a parahydroxyl group, and it's got this group and it's got this group. So I don't know yet, do I want this negative, do I want this positive, and what happens to this OH group? So tyrosine, just to show you, here it is. Okay, oh, and it's got this extra carbon there. I think I forgot that. All right, so basically what I'm saying is that if we're at a pH of 9, then this is going to be deprotonated because the pKa is much lower. This one is going to be about the same, so it's going to be either protonated or not, 50-50. And then side chain has a pKa of 10.7. So let's say that's almost 11, even though it's logarithmic. So if it's a little bit more, pKa is higher than the pH. I like to think of it the other way around. I look at the pKa and then I look at the pH and I say, oh, the pH means that we're in a more acidic solution. So more acidic solution means it's protonated. So at pH 9, we're likely to have this hydrogen, but at pH 12, we're not likely to have that hydrogen. All right, so that's the most likely form. And again, here, this could go either way. It could also be neutral in some of its 50-50 at approximately same pH and pKa. So one thing that's useful is to calculate the pH at which you have achieved the greatest concentration of zwitterion. So where can you get this to be balanced out and be the most negative here and positive here? So in order to get to that pH, we do a calculation for the PI. Okay, so PI calculation is what pH do I need to get the most zwitterion? So to get that number, it's usually pretty easy. You take the basic site and the acidic site, and take the pKa's of those, and you divide by 2. And you'll see most of the PIs are, as long as there's no other acidic side chain, it's going to be 6 or 7, right? Because you have pKa of 2 or 3 and 9 to 10, so the average of all that. Now, if you have an acidic or basic side chain, then that has to factor into the calculation. So in that case, we usually just take, if you have a basic side chain, we take the 2 basic ones and we take the average of those. All right, and same with this one here. So I have carboxylic acid. So I know I'm going to end up with something pretty low pH, which means this is definitely going to be charged. So I don't have to worry about that. What I'm worried about are these guys. So that's why I use those in the calculation. So why that's useful is it sort of gives me an idea about the overall amount of charge on the molecule. So if it's in its zwitterionic form, it's sort of got charges on it. Is it such that it overall has a net positive charge, a net negative charge, or is it balanced out? So at these different isoelectric point values that were calculated, how you could use those values is see at a given pH, let's say pH 6, what's the amount of charge you might have at that pH. So for example, for alanine, which is at that pH, the pi is equal to that pH, then you're not really going to have negative or positive charge. You're going to be evened out. But if you're at a pi greater than the pH, then you're going to end up with something with more positive charges on it. So that means because your solution is more acidic than your pi, so that means you're going to have mostly things in their acidic form, which tends to take on positive charge, especially for amino groups, right? So this means a positive, mostly positive charge, and that will be attracted to negative charge if we were to run a gel electrophoresis with a negative terminal and a positive terminal. Okay. Likewise, if we have glutamic acid, okay, and it's got pi that's or pH is more basic than the pi, then I'm going to end up with more negative charges, and that's going to be more attracted to positive end or terminal on a gel. And often what people do with these gels is they take a protein and they digest it or break it into smaller pieces, and based on those amino acids and what charge they are, they flow through a gel at different rates. And so you can separate proteins this way. And by the way, one way that you can see where the proteins travel or where the amino acids travel is by attaching a dye to them. So you mix the amino acids or peptides with a dye. And that dye, for example, reacts with proteins. So you could use them for fingerprinting. And so that's one technique that forensic scientists use is at a crime scene. If they're looking for fingerprints, they'll spray some of this ninhydrin, which is this compound, and it will react with the amino groups and bond to, the dye will bond to the amino acid.